Cultivation of viruses, Part 4. In this video, we will have a look at in vitro cultivation of viruses. A cell culture is prepared by encouraging cell growth outside the animal or plant source. So, although lab animals and embryonated eggs are very useful for isolation of certain viruses, cell cultures are the only system for viral isolation in most labs. Cell culture systems have, in fact, been of immense help in the growth of the discipline of animal virology. To prepare a cell culture, tissue is taken from a healthy animal. It is then fragmented by mincing, and the cells are dissociated with the help of the enzyme collagenase. The cell suspension is now filtered and washed, and the cells are separated by the process of differential centrifugation, after which the cell suspension is briefly treated with trypsin. This is to detach the cells, and then the cell suspension is placed in a flat bottomed glass or plastic container together with a suitable nutrient medium. Now, this nutrient medium includes a range of salts, vitamins, hormones, and other growth factors, amino acids or proteins, glucose, antimicrobial agents, a buffering system, a pH indicator, some non-specific sources of nutrients such as fetal bovine serum. All these are components of this nutrient medium. So this medium, as you may have heard just now, it has a wide range of nutrients which will support the growth of cells. But it will also be vulnerable to a wide range of bacterial contaminants. There are antimicrobial agents in this medium, but even then, the cells must be grown under stringent aseptic conditions. The cell suspension that has been introduced into this medium, now those cells will start attaching to the surface of this container. So the surface is, as earlier said, it can be glass or specialized plastic. And these cells, we call them as anchorage dependent cells. That is because they need a solid surface for attachment and growth. And the surface of glass or plastic, this is providing it with a surface. Now what happens is that on this surface, the cells start to divide and they start to grow across the surface. They undergo mitosis and they form a sheet on the surface of this glass or plastic as they grow. So there is a single layer of cells or a monolayer of cells that is formed. And this layer of cells which is formed, it will keep on dividing, and this entire surface now will be covered with cells, and it is called confluent. So this is a situation where the cells are covering the entire surface and they are in contact with each other. And when the cell contact happens, the cells will start dividing. That is called as contact inhibition. And due to contact inhibition, the density of cells doesn't become too high. So far, what has been described is actually called as primary cell culture. So here is lung tissue And as the procedure has already been mentioned, the tissue is taken and treated with enzymes, and finally you have a cell suspension, and this is introduced into a flask, or here in this case a Petri plate, and you find that the cells are spreading until a monolayer of cells is formed. And this is actually called as a primary cell culture, where directly the culture is developing from living tissue, and it may contain different kinds of cells. Now, in order to prevent contact inhibition, because once contact inhibition occurs, the cells will stop dividing. So what can be done is cells from this primary culture, it can be transferred to another vessel, which has fresh growth medium, and this is called a secondary cell culture. And this can also be maintained by periodically reducing the cell density. You can pour off the excess cells, add fresh medium to provide space and nutrients, and the cell growth can thus be maintained. Now, this primary cell culture cannot be cultured more than a few times, and you may need to obtain fresh tissue from animals on a regular basis. For example, in this case, the primary cell culture is directly from lung tissue. There is another terminology called diploid cell lines. Now, these are certain cell lines that are developed from human embryos. But unlike in the case of primary cell culture, the diploid cell lines can be maintained for around 100 generations. And they are used for culturing viruses that require human host cells. Now here you have what is called as a continuous cell line. So what is this continuous cell line? Now these are cells that have lost the normal constraints of contact inhibition and mortality. So these cell lines are actually derived from tumor cells, from transformed cells or tumor cells, and these can be subcultured many times. They can be grown indefinitely, and so they are immortal. They are said to be immortal. Now here you can see a representation of tissue which has both normal as well as transformed cells. When this is cultured, the normal cells, they are forming a monolayer as they exhibit contact inhibition. But the transformed cell, that these do not form a monolayer because they do not exhibit anchorage dependency, which means they actually do not even need a surface to get attached to. They can even grow in a suspension, and they have lost contact inhibition. As a result, they can grow in piles or lumps like this, resembling small tumor growths. For example, the strain of cancer cells called Henrietta Lacks strain or the HeLa cells, they are examples of this kind of continuous cell line. Now, cell cultures vary greatly in their susceptibility to different viruses. So what we have made the cell culture for is now we are going to inoculate those cells with, we want the cells to be host cells to viruses. For a particular virus, those cell cultures should be used that are most sensitive to it. Now, suppose there is a particular virus of interest and that grows in the oral cavity. So what we do is we have to take a throat swab and that is placed in a vial. So a throat swab is taken and that will be placed in a vial in an appropriate medium. a viral transport medium, and then it will be sent to a lab after being taken. And it has to be sent as soon as is possible. And upon receipt of this viral sample, the sample will be inoculated into these different uh, flasks which have appropriate culture media with the cell cultures. So depending upon the specimen. And then these flasks with the inoculated viruses, they will be incubated at 35 to 37 degrees Celsius for a few days. That is so that the virus grows in the infected cells. Now, cell culture, after this time, it will then be inspected under low magnification inverted microscope. And what are the changes in the cells themselves due to the viral infection that will be looked at. So you can see here, this is the cell culture before infection and after infection. So what are the changes that are taking place in the cell culture due to the viral infection that is known as cytopathic effects or CPE? And these include loss of adherence to the surface of the container, changes in cell shape from flat to round, shrinkage of the nucleus, even the cells may shrink, vacuoles in the cytoplasm, fusion of cytoplasmic membranes. Now, in fact, here you can see in this picture, this is called a stranding of the cytoplasm. This is also a cytopathic effect. Then presence of inclusion bodies, that is within the cytoplasm, you can see some kind of extra structures, or a complete cell lysis could also occur. So once you see some of these effects, you know that the virus has affected the cells which are being cultured. So these viruses then need to be harvested and purified from the cells of the monolayer or the cell culture medium for further research and investigations. So what are the advantages of cell culture? So whole animals need not be used, and cells keep on growing in the cell culture, and they are a renewable source. You can grow different kinds of cells, including human cells, and inoculate the medium and the cell culture with viruses, appropriate viruses. Cells can be grown in various containers, and the numbers depending upon requirements. And if they are surplus cells, they can be stored indefinitely in liquid nitrogen. And yet there are disadvantages too. What are the disadvantages? You need to maintain sterile, aseptic conditions. The chances of chemical and microbial contamination are very high, and experience is needed in maintaining these cell cultures. The capital investment which is needed to set up a cell culture facility is very high. And due to rapid growth rate of cells in artificial culture, there is a chance that genetic variation may be introduced into the population. Now, in spite of the success of cell culture methods, however, there are still some viruses that have never been successfully cultivated in cell culture. So with this, we complete the topic on cultivation of viruses. Thank you.